How about now? There'll be like a 5-minute delay in whether this works or not works.
Yeah, we can hear.
Okay, great. Really quickly, for the people online, one of the things that you should take away from this talk is that proteins are dynamic complexes. They're changing their conformations, the partners with which they associate, and that's really what allows them to function. The second thing is that we typically look at proteins in isolation, and I think one of the most exciting things happening in structural biology and cell biology now is electron tomography, which allows us to look at complexes in a cell. And that's something I'm showing here. This is a bacterial cell, actually called Mycoplasma pneumonia. And what I've done here is I've highlighted ribosomes within that cell, these distinct structures, and so you can actually place structures of interest into a cellular context. Okay, so I'll sort of talk to you about some of the deep learning approaches that we've been using in combination with data from cryoEM and cryoET to try to understand how heterogeneity drives function. Okay, and I thought I would start by just sort of, again, articulating how important structure is in understanding function. So, decades of work have really highlighted, the importance of high-resolution structures and understanding how macromolecular machines work. I have a couple examples here. The ribosome we'll talk about extensively. It drives all protein translation in your cells. The proteasome we'll talk about, maybe not at all in this talk, but something my lab is interested in, really drives turnover of proteins in the cell. And there's a bunch of other complexes. All of these things are coming together to form functional complexes. What structural biology has thus far not been that helpful with is understanding how you build these things. Right? So we can look at the terminal complexes and learn something about them, but it's been very hard to understand how the cell takes a pile of individual components and properly folds and orients those relative to one another to give rise to a functional machine. And I don't know if he's watching, but I have a 4-year-old son who asked me to incorporate this slide, which is why you see an excavator. This is the obsession of all 4-year-olds, as far as I can tell. So hi, Will, if you're watching. Okay, so how do you take these things, fold them, and orient them to give rise to a functional machine? And then if the cell state changes, so you move into a new environmental condition, maybe the machine becomes damaged, kid steps on it, whatever it is, can you disassemble it and then recycle those components? And how do you do that? How do you order those processes? And this has historically been really a hard space to access, in part because it's extremely heterogeneous. Right? If you have to put together a whole bunch of different parts, almost by definition, the intermediates are going to be heterogeneous. There's going to be some that have component A, some that have component B, some that have both, and biochemically isolating any of those things is challenging. Okay, but if we could do that, we could start to ask questions about, how does the cell's ability to assemble and disassemble these things change by some perturbation? So those could be environmental stresses, it could be disease predisposing mutations, and there's a lot of data that says that both these environmental stresses, age, and disease predisposing mutations impacts the cell's ability to build and degrade these complexes, and that's linked to a ton of pathology. Right, so if you could understand this space, you could have big impacts on human. These are the types of questions one might ask. If you think about the cell as a factory, you could ask, if you want to build a car, is there one way to build it? Is there just an assembly line that always goes down the same way? The chassis gets built first, then the wheels go on, then the bonnet or whatever it is. Or is it flexible? Do you have sort of flexible manufacturing where you can build different components in different orders? Do you build whole subcomponents? So like, do you build a whole engine block and then drop that into a chassis? Or do you sort of build things in situ, one screw at a time? What happens if you block a step? So if somebody doesn't come to work, do you shut down the factory, or do you figure out ways to route assembly flux around that blockage such that it can be recovered once that worker comes back? And then how do you recover from errors? We know that these are not perfectly faithful processes. If you put something in the wrong order, in the wrong orientation, do you have to start over, or are you able to fix it in situ? Okay, so how have we tried to analyze these questions historically? And most of the structural data that is in the PDB that folks in design are leveraging comes from X-ray crystallography. And I just wanted to spend a quick second how people think about this data and maybe how it impacts the design paradigms that you guys use. So the fundamental problem with imaging biological molecules is that they get damaged by radiation, whether that's from X-rays or from electrons. Right, so if you're going to take a picture of it, you're going to irradiate it. That radiation is going to cause some sort of damage, which means that you have to limit the amount of dose that you have on any particular particle. Okay, so crystallography is great because now instead of irradiating one particular particle, you can irradiate through a crystalline lattice, billions of molecules. You can spread that dose across, but because they're all in the exact same orientation, the same conformation, which is defined by the crystal lattice, you can sort of integrate that signal. All right, so you're basically physically constraining the molecules such that you make the physical averaging process possible by being able to irradiate through the crystal. The downside is that I told you at the beginning, proteins are dynamic. They're dancing on this complex energy landscape, and now we put them into a jail cell because we want to be able to image them. And if you think about this in terms of like docking a small molecule, I think about trying to feed a kid, right? This looks like a relatively trivial problem, but the dynamics, the dynamics are actually sort of challenging to get the small molecule into the right orientation, right? There's a whole bunch of things going on there, and if you really want to be able to dock the molecules, you need to be able to consider all those dynamics. What's that? Is what? Oh, that was my audio. Yeah. No. Baby. No, that was actually me in the background saying baby. Okay. For this audience, I threw these slides in and just okay. So I'm an experimentalist by training. I think that experimental data is super important. I imagine this audience is like, Joey, we have AlphaFold. Like, why are you going to determine structures? And AlphaFold is transformative. It is. I'm like the biggest fan. But I want to just highlight sort of what problem this solves and contrast that with the problem that I'm trying to solve. So, AlphaFold solves this problem of going from primary sequence to a folded structure, and it's absolutely remarkable if you compare the experimental data to the prediction. It's incredible, right? Like if you have a relatively small protein, something less than 30 kilodaltons that has a globular fold, do it, right? Like that's the right approach. So what does this look like? All right, so this is drawn to scale. This is some protein of interest. It's something on the order of like 15 or 20 kilodaltons. It's folded extremely well. It's well predicted by AlphaFold. The complex that my lab works on primarily is the ribosome, and on scale, this is the ribosome. Right? It's dominated by RNA, which is hard to predict. It's extremely dynamic, and it's massive. Right? This we can't currently predict. What's going to allow us to be able to predict this? Like I have full faith that deep learning is going to allow us to predict these sorts of structures in the future. But right now we are very data limited to be able to train models to do this. Right? We have a shitload of this from crystallography and relatively little of this. How are we going to get that data? I posit that that's going to be through cryoEM. And so for folks that haven't seen this before, this is the way the technique works. So this is an EM grid. It's typically some metal grid, these grid bars. Laid across the grid bars is a very thin layer of amorphous carbon. And using photolithography etched into that thin layer of amorphous carbon are small holes. And across those holes, you can suspend a thin layer of vitreous ice containing your protein of interest. Okay, so this is on the order a couple centimeters, 3 centimeters across. And then this is this very nice video from Twitter. Wow. It sort of shows you zooming in first with light microscopy and then with electron microscopy. We're going to go deeper and deeper into this grid. Okay. Now we're in the EM regime, that low mag. We're going to go further and further. Now you can see the same amorphous carbon with this pattern of holes in them. Inside of each one of these little holes, that's where this thin layer of vitreous ice is going to be. And then suspended within that vitreous ice are going to be our protein complexes of interest. Slowly get to. And then we're going to low pass filter the image. And you start to see these individual molecules. All right, so this is a single molecule approach. We can actually see individual molecules. Each one of them is this unique, beautiful snowflake, each in its own conformation, but they're really shitty snowflakes, right? The signal to noise is terrible. It's very hard to see each one. And this goes back to this fundamental problem that we can't dose the sample, because if we dose the sample, we'll damage the biomolecule that we're looking at. So we have to apply extremely low dose. And so the shot noise is through the roof. You know, the SNR on these things is like 0.1 or 0.25. It's less than 1. Super, super low, but there is information there. And so what we end up doing is using that, using these original micrographs, we can then pick individual particles. We can extract from these particles on the order of hundreds of thousands to tens of millions of individual particles. And each one of these, again, are pictures of single particle images. And because we haven't constrained these things using a physical crystalline lattice, it provides the opportunity for us to find rare but biologically informative states. We could quantify the relative abundance of these states. And so the dream is that you could sort of compute an energy landscape to give yourself a sense of the distribution of states that were adopted. If you could do that, then you could do things like discover structural independencies. And you could test hypotheses that relate some experimental condition to structure, right? So instead of getting one structure, you get an ensemble of structures. You change the condition and ask how that ensemble changes. Okay, this is fundamentally a computational problem to figure out how you could leverage this low SNR data to go after these opportunities. And so the way that people have historically done this is one of two things. They will treat the particles as though they're structurally homogeneous, and they're basically going to try to infer a voxel array. So that is this box, where each position in the box, you have some E in density that tells you the density of sort of signal from the heavy atoms. You're going to infer this from the underlying particle images. And in some ways, you can just think about that as averaging. And inherent in averaging is this baseline assumption that the particles are structurally homogeneous. We know that that's not the case. And so you could relax this constraint a little bit and say, oh, there's not one structure, there's two structures. And so now, instead of inferring one voxel array, I'm going to infer two voxel arrays. And for each particle, I'm going to assign it to bin 1 or bin 2. And if you have a bigger GPU, you could do this with 4 or 8 or 10. But the combinatorics of assembly problems are not in your favor. Okay, so let's just think about a couple examples of this. Imagine that you just have two components binding to an RNA. This is the RNA as a black box. The red component or the orange component combined. There's 4 states, completely unbound, red, orange, or both. We have 4 components. We're up to 16 states, 6 components, 64 states. The ribosome, the particle of interest for me, it's preposterous, right? You're not going to do a K equals 1 billion state classification. Just not going to work. Okay, and force biology is likely not going to occupy all these states. There's going to be cooperativity. There's going to be some ordering, but formally, all of these states are possible. Okay, so this was sort of the mental hurdle when I started my group that we were trying to overcome, and we were trying to think about ways that we could reframe this problem. I'm going to, in the interest of time, just jump ahead a couple slides. We started late, and I don't know. We can go a little. Okay. If you want to hear more detail, more exposition on why I think this is important. All right. Fair enough. Sorry, people on Zoom, you didn't get to vote. Okay. All right. You might respond to me and you might say, well, Joey, I don't care about assembly. I know I have a single polypeptide. There's none of this sort of combinatorics about compositional heterogeneity. I just think that it's in different conformations. Okay. So let's imagine that case. Let's say that you have 2 states. You have a state where this domain is up, and you have a state where this domain is down. And it could be that this is a highly cooperative transition such that the intermediates are unpopulated. And if you did this and you used a discrete K equals 2 multi-class refinement, you're going to do a great job. You're going to get high resolution structures of those 2 states. Now, this is a continuous transformation. And so we know that there must be at some low level populated intermediates because it has to progress from state 1 to state 2. These are so small that, Joey, I don't really care. I'm not going to be able to learn anything about them. Fine. What if those are a little bit more abundant? Now you have 2 choices. You can either throw this data away, ignore it, do a 2-state classification, and get 2 high-resolution structures, throwing away data, or you can try to use all your data, in which case these are going to blur out one of your states. And in the extreme example, you have 2 choices. You get 2 low-resolution structures from which you can't interpret anything, or you throw away 90% of your data and you get 2 high-resolution structures. And if you guys go and read CryoEM papers, buried in a supplement, there will be an extended data figure 7 that shows their cryo-EM processing workflow. And they will typically say we extracted on the order of 2 million particles, and here's the structure that was representative, and there were 100,000 particles that went into it. And don't worry about all those other things. Maybe they were denatured, could be, or maybe they just threw away things that didn't. Okay. So this is the conundrum. The way that we started thinking about it was that we started thinking, well, instead of using the data to try to infer the voxel array directly, let's think about trying to use the data to just learn a mapping function. So this will actually probably come more naturally for this audience than most times I give this talk. So We'll see. But it's a little bit counterintuitive. Instead of learning this, I'm going to learn a mapping function where the mapping function is parameterized by some 3-space Cartesian coordinate, and its job is to take that as an input and output the density in 3 states. Okay, so if you could learn that function, the way you would use it is you would just rasterize through your box, and at each position you would predict the EM density, and once you've rasterized through this whole box, you're going to get back the EM density. Okay, we're back where we started. We have one structure for one data set. If we have this formalism to think about this as a function, well, functions can take all kinds of inputs. But now let's, instead of just parameterizing this on the 3 Cartesian coordinates, let's give it an extra input of a discrete class, k. If we could do that, then maybe we could give it a continuous variable, which might be some position along a reaction coordinate. If we do that, and we're sort of dreaming the dream, then maybe we could give it some position on some high-dimensional energy landscape. And conditioned on the position of that energy landscape, we could get that into volumes. How would one learn a mapping function like this? So what we began thinking about was that approximation theory tells us that if we can provide the right inductive bias and the right types of data, we should be able to learn the mapping function. And so this is sort of the formalism that I laid out to Alan Zhang, who was the first student that joined my lab. And I would say that the key to making this work, and in general to making ML approaches work, is finding the right person that can figure out the right tricks to make these work. This is really just a monument by Alan, who now started her own group at Princeton. Okay, so how did Ellen figure out how to learn this mapping function? Broke this into 2 parts. The first part is exactly what I just told you. There's some details here which I'm happy to discuss about how you sort of convert from these Cartesian coordinates to a code that's learnable by an LP. The basic idea is that you have this, what we call a decoder network. It's going to predict the density at some position. We're going to rasterize through this whole slice, produces a model slice. We'll compare that to the input image. The difference between them we'll use to update the neural network weights on the decoder. So we'll just pass the data over and over and over again until it can predict good model slices. To handle heterogeneity, we set this up as an autoencoder. So there's an encoder network in front of this. It's going to take these 2 images, map them into a low-dimensional space, and then conditioned on this latent code, the decoder will give us. Okay, so the way that one would use this is that you would take what we call CryoDragon, this variational autoencoder, pass a given data set through it, something like 10 to 50 times until the networks are trained. You'll do one final pass where all the particles get mapped into this low-dimensional latent space. And here I've just colored them by particle density. So you can see there's on the order of 6 major states, but there's actually populated intermediates between. It's an energetic generative model. Wherever you want in the latent space and generate density maps. And so you can do things like, what are the major discrete classes? You can get those back. But I think more excitingly, you can sort of walk along the data manifold and you can start to see these continuous changes. Okay, a couple ways that we tested this. I'll go back to this example of a continuous conformational change, again, starting in this up position and moving it to a down position. We could simulate this data. We could talk about, if people are interested, the challenges with simulating cryo data. But whatever the case, we did these simulations, and then we have a nice ground truth of what the reaction coordinate is. And so we can either have something completely continuous, something highly cooperative, or something entirely cooperative where the intermediates are not even populated. Put that through CryoDragon, and then we can look at the correlation between the true reaction coordinate and the learned reaction. And then, of course, this is how it learns, right? It starts with sort of the initial model is basically that it's a unit Gaussian, and then it's going to learn to separate out these distributions into states. And then we can sample along them, and we get back this continuous conformational change. Okay, this works on real data. So this is the spliceosome. So it performs splicing in eukaryotes. If you look at so these are 2D class values. So this is taking a whole bunch of the individual particles, aligning those that were viewed from the same projection angle. What you can hope to see is that there's high resolution features for the portions of the molecule, and then there's these blurred features in the distal parts. Those blurring typically come from motion that's being averaged across these classes, and you also see that in 3D. This is a local resolution plot. The blue stuff is high resolution features, and the red is low resolution. So it turns out that this is sort of like a very nice model data set for people to try new algorithms on. That's in part because if you know a lot about the spliceosome, you can draw masks around various regions. You can refine those independently and push everything to high resolution. Yeah. Yeah, so one of the challenges is that you need to do two things for cryoEM. You need to figure out what angle you took the picture of the molecule from. So we call that the pose, or it's a projection angle. Then you need to figure out the underlying structure of the molecule. It turns out for all these algorithms, they really need some domain that is rigid enough to do the alignments to get that initial pose. And then once you have the pose, then you can start to see these blocking. So things that are purely IDRs, nobody can touch. We don't know anything about the spliceosome. I don't know where to draw the masks. What I think is really neat about this approach is we can just feed it the data. One more time. We can just feed it these data sets, and if we walk along the data manifold, now we actually start to see these motions that were blurred out previously. There's no masks applied here. There's no expert guided intervention. We're just letting the data define the sort of motions that we can see. Okay, so we've had this tool. My lab's interested in ribosome biogenesis, and so I'm going to tell you a story of sort of how we're using this and what we think we can learn about how cells assemble ribosomes. So this was a really nice collaboration between Laurel Kinman, fantastic graduate student in my lab, Joaquin Ortega at McGill University, and a graduate student also super competent in history. The goal here was to try to understand how a particular assembly factor, happens to be called KSGA, how it drives assembly of the small ribosomal subunit, highly conserved. We know that it has some activity on methyltransferase, which is involved in installing a methyl group on the RNA that helps translation. But there was all this data that it was involved in assembly, and nobody really understood why or what it's. Next here. Okay, so here's the experimental design. We're going to grow cells in the absence of KSGA. The idea is that they should accumulate ribosomes, which are the substrates for this factor. We're going to split those into two groups. One will treat with the factor, and one is untreated. And we're going to image each one of them independently. Then we'll use this tool CryoDragon to compare the structural ensembles between them. So we first consider the KSGA treated sample. This is the learned latent space. You can see that it's highly featured. There are, very highly populated states. And just give yourself a sense of scale. When a domain like this is missing, that's something on the order of 300 kilodaltons. So that's like 4 or 5 proteins and a huge swath of RNA. So this is extremely heterogeneous. But to the question that was asked earlier, it's using the body of the particle to drive the alignments. So we can see things with KSGA down. We can also see things without KSGA. You can see highly immature, highly mature. I motivated also by saying we don't want to just look at 4 structures. We want to look at hundreds of structures. So we have this generative model. And so what we can do is we can just sample throughout latent space. There's a bunch of different ways you could do this sampling, but you can generate on over 500 or a few thousand different structures. And then you have 500 or a few thousand different structures. And then what, right? It's a pain in the ass to build models for one structure, particularly something as large as the ribosome. And I'm very good at motivating my students, but I cannot convince anyone to build models for all of these and interpret them. So we try to take a little bit of a simpler approach to think about looking at these ensembles. The basic idea was, well, we have the atomic model for the 30S. So what we can do is just dock that in, and then at every position that we're interested in, be it our ribosomal protein or an rRNA helix, we can just ask how much of the density that we know should be there when it's natively folded do we actually observe. Okay, so the way that would work is, here's 2 different maps that were sampled. You can see that they both have this helix 15 well occupied. One is lacking helix 44. You can see that the model being there, but not the rest of the density, and one of them has it. Again, then we can parameterize that at what we call an occupancy that scales from 0 to 1. And now we can do this across all 500 or 1,000 maps. So that's each one of these different rows is a different map that was sampled by CryoDragon. And each one of the rows is a different element, be it an RNA helix or ribosomal RNA, or ribosomal protein. Okay, so what can we actually learn about assembly by looking at this? Here's a couple of examples. So we can find classes that are extremely mature. Trust me, this is what a mature 30S ribosome looks like. We can see things that are highly immature. We can find cases where the head, that's this domain here, gets built first, the platform is absent. We can find other cases where the platform is built first and the head is absent. So this tells us that they're incomplete by parallel pathways. Right? If it's not obligate that you put in the head and then the platform and then mature. You can do it in either order. You can ask things like what kinds of particles can KSGA bind to. So we just basically look at this matrix, say, give me back all the particles where KSGA density is present. What do you see? You see KSGA bound to extremely mature particles, things lacking the platform, and things lacking the head. And that tells us that really KSGA is recognizing the local structure on the particle, and it's less sensitive to these distal elements. Yeah?
So you see that there's those two different pathways. Can you say anything about the flux?
Yeah, it's a great question. So we certainly can't say what's kinetically preferred. We have exactly one point, right, which is when they were frozen. What we can say is that that is something like an instantaneous distribution of particles. It's not at equilibrium. And so what you can do is you can say, like, what's the frequency with which I see the headless with platform versus the platformless with head? And you can start to wave your hands and make arguments about maybe one being preferred because you see more occupancy in that pathway. But we don't have the kinetics, and so being able to say that authoritatively is challenging. All right. Next question we asked is can we use this to now enumerate binding dependencies between structures? And what I mean by that is, since we have the occupancy for each element for each one of the particles that we have a structure for, we can just look at correlations between them. So you could look at things like this one is important, this helix 484 and this protein KSGA. You can actually see that they might sterically occlude one another. And so perhaps it's not surprising that they're mutually exclusive. Whenever KSGA is high, there's no H44. Whenever H44 is high, there's no KSGA. You can apply this now across all of the RNAs and all of the ribosomal proteins, and you find some interesting patterns. So you find things that look like this, where S3 is only present, so its occupancy is only high when H35 is high. But the opposite of that, is a contrapositive or some variant of that word, that is not true, right? So you never see particles where S3 is high without H35. So now you can put a directionality between these different elements. That S3 must be downstream of H35. You see that for many elements, but not all. There are some that bind independently. You can have high H23 without H35 and vice versa. So you can take all these interdependencies and use that to build up a graph that tells you what does the assembly look like. And this is, at least for someone that's worked on the ribosome, this is extremely satisfying. So I don't know if anyone has ever heard of the Nomura assembly map or the Nearhouse assembly map, but these are sort of seminal experiments done in the 70s by these like incredible biophysicists. And the way these experiments work was they purify all the ribosomal proteins, something on the order of like 30 to 80 proteins. They purify the RNA, mix them together, and they could reconstitute an active particle that was fully assembled. So that's heroic. The incredible thing about these guys is they convince people to now repeat that experiment, but withhold one protein. So they pull one out, and then they ask, what kind of particles assemble? When you pulled out particle A, or sorry, protein A, maybe B and C don't bind. And so you can say that B and C are downstream. They did this systematically across the ribosomal proteins, and they could build up these interdependency maps that told you what the thermodynamic couplings were across all the ribosomal proteins. And what was so cool about this analysis is that much of the Nomura assembly map just sort of fell out of looking at the landscape of particles that were on our grids. The other thing that's cool about it is that now we could start to put the RNA helices into this map. So you could start to see how protein binding events were linked through RNA folding events. You might have a protein bind that creates a docking site for an RNA helix. Once that RNA helix is there, a new protein binding site is event, and that explains the interdependence between the first protein and the second protein. It's mediated by the docking of an RNA helix. To help us really explain how that works. Okay, so I told you a bunch of things about assembly generally, and we're told you about what KSGA does. So we started to look at the other sample, which in theory should be the substrate of KSGA. We expected this to be even less mature than what we started. And we were really surprised, it doesn't matter how we parameterize this, the particles grossly appear more mature. So the substrate of the assembly factor is more mature than the product of the assembly factor. That doesn't sound like an assembly factor. And so this was a bit of a confusion for us. And we started thinking about what regions KSGA might be interacting with, and we knew that it bound this sort of mutually exclusively with helix 44, and it turns out helix 44 is really involved in a bunch of biology of the ribosomes. So we started focusing on that. And we did that by drawing a mask around helix 44. And now for every one of our particles, we could go and generate a structure to look at what's the conformation of helix 44. Okay. And if we take all of those structures and do principal component analysis on those volumes, we can now walk along the principal component. And what we saw is that there's sort of two major conformations. There's one where the helix is sort of tucked into the body. There's another one where the helix is released from the body and sort of flayed out with this. And it turns out in the late 60s and early 70s, these two conformations were described or were predicted by chemical probing experiments. This one is inactive, able to do translation, and this one is active. So we were sort of curious that we have to be able to find these two conformations in our data sets, and what it looks like if you traverse across them, it sort of starts in, it gets played out, it'll come back. So we had these two different conformations. We could ask, what is the relative abundance of these two conformations in either the KSGA treated or the untreated samples? And so if you look in the absence of KSGA, you see both conformations. You see the active conformation here and the inactive conformation here. But if you look at the KSGA treated, the inactive is completely pruned away. Right? So the precursor particle, the substrate, has a ton of inactive things. You treat it with KSGA, it grossly looks less mature, but the things that are there are all mature particles. So this is actually sort of consistent with this notion of kinetic proofreading that we see in a bunch of other enzymes. You can have pathways that give rise to sort of malformed structures, and those malformed structures are specifically recognized, disassembled, and then given another chance. And that's sort of the model that we come up with out of this. Is that you have precursor particles, they partition, some can mature on their own, but many of them form this inactive helix 44. KSGA comes in and recognizes this specifically. Upon binding, it displaces helix 44. That leads to a bunch of other computational changes I didn't describe for you guys today, including uncoupling of its head. That's why they grossly look less mature. But these particles have a new chance to assemble and assemble in the proper conformation. Sort of increase the overall assembly fidelity by going through this proofreading step. Okay. There's been a bit of sleight of hand in everything that I showed you, and that is that we grew these cells, we lysed them, and not me, but somebody spent 4 or 5 days purifying the particles. They put them on grids. We looked at the distribution of conformations, and we said that's what was in the cell after we went through all these columns, right? That it didn't re-equilibrate to some other state that's completely uninformed. This concerns me that we have to make that statement, but that is generally what structural biology does. And so what we'd really like to know is how are these machines assembled in situ? And in the last probably 5 years or so, this approach called cryo-electron tomography, I think, is going to give us the ability to start to ask questions about how things like ribosomes are assembled in situ. The way this works is that you take your same cryo-EM grid, you now either grow on it cells or you deposit cells on it. Turns out those are too thick to image typically. So use a focused ion beam and you're going to cut a lamella into those cells. So a very thin window that is sufficiently thin to use transmission EM to look through it. You're still so thick that you have a bunch of particles on top of each other, and so you have to collect a tilt series to disambiguate the z-axis of those projections. And so you collect this tilt series, which results in what's called a tomogram, which is really a 3D volume of some subsection of the cell. Within that tomogram, you can pick individual particles, things like ribosomes, and then historically people would do the same sorts of averaging approaches to mix all those subtomograms together to increase the resolution, and then they'd actually see these structures now mapped to their cellular location. Okay. Same problem that we have with single particle, you're averaging and you're assuming structural homogeneity. We were curious if this same CryoDragon framework could work in a tomographic setting. And so the difference is, instead of having a single particle image for each particle, we now have a full tilt series. So this is sort of a multi-view version of the same thing. But in theory, this should work more or less the same. It turns out the details of this make that not trivial. And so Barrett Powell is this incredible student who actually started as an experimentalist, had never coded in his life. The pandemic hit, and he's like, I don't want to sit on my hands. And he just like learned to become a coder on his own. Really remarkable. So proud of him. So he put all this together, figured out all the details to actually make this work. Okay, so we can talk about the details in the Q&A if we're interested in exactly what you have to change here, but there's a bunch of small details that end up mattering a lot. The way that this works is, or the way that we tested this, was to first start again with simulated data. We took 4 different assembly states of the ribosome. We simulated tomograms of those, put them through this TomogrDragon program, and then asked them to just classify them and see that 4 groups come out. We can generate structures from those, and they nicely map to the ground truth that was the input. And if we look at the confusion matrix, it's nearly perfect in its assignments. We can also look at continuous conformational changes, so we can simulate, again, tomograms of particles undergoing this sort of weird rotary motion that the tryptase undergoes. And if we look at the data manifold for those, you can see now it's actually nicely organized by the ground truth reaction coordinate, going from purple to yellow, and we can sample along it, and we get these nice rotary motions. Right, so it works well with simulated data. What about real data? This is an HIV virus-like particle. So it turns out, this is sort of surprising me, but you can take the primary protein for HIV, it's this GAG protein, put that into a test tube, and it'll assemble into these sort of spherical or nearly spherical structures called a virus-like particle. I don't know if you guys can see this, but they're sort of decorated by a couple rings. Those are the main things. So from these VLPs, you can collect tomographic data, and we can reconstruct what looks like it turns out to be a hexamer of each of those. Apply that to, and I should say that this basically recapitulates the sort of structure that everyone determines when they look at this by tomography. But if you look more closely with TomogrDragon, you now actually start to see that there's heterogeneity. There's a whole other domain that's a little bit below the primary domain that everyone sees. That's encoded in the polypeptide. Everyone knew it was there, but everyone thought it was just too flexible to be able to see. And so I think this is back to your initial question. Flexible things we can see so long as you can align. And now we were able to start to see these. Turns out this is the domain that actually binds to the nucleic acid eventually assembly. What's cool about this is you can take these classes and map them back to the original VLPs in the tomogram, project them on the sphere, and you can start to see why the spherical lattice gets broken and why it doesn't form a perfect sphere. It turns out it's these nucleic capsid or these In-situ domains that are binding nucleic acids that sort of co-cluster on the structure, that sort of breaking the symmetry. Okay, what about in actual cells? So this is a beautiful data set that we didn't collect. William Mohammed's group collected a few years ago now. And so they took Mycoplasma pneumonia. It's a very, very small bacterial cell. What's cool about it is it's so small you don't have to do this fib milling. You can just image it directly. So it's a perfect imaging system sort of test algorithms on. And once you image it, it's a little hard to see here, but there are actual ribosomes throughout there. If you denoise the micrograph lobe, you can see them nicely. You can extract those particles. You can extract those particles and generate a high-resolution reconstruction. TomogrDragon can also generate a high-resolution reconstruction. And I think the most striking feature about this data set is that they had treated it with a small molecule chloramphenicol. This is an antibiotic that binds to the ribosome and blocks translation. And we can actually find density for chloramphenicol. So this is a small molecule therapeutic bound to its known target, imaged directly in a cell. It's a beautiful statement to make. It turns out it's an extremely abundant target. It's an extremely large target. There's all these things that make it possible, but I still think it's incredibly exciting that we can see small molecules on their real targets. Okay. There's not just one structure there, of course. If we look by TomogrDragon, we can see a bunch of different structures. I'll take you through a few of them. This is elongation factor T, that's sort of the primary elongation factor that drives translation. We can see particles where EFTU is bound, we see particles where EFTU is absent, that red dot there, versus it not being there. And sort of interestingly, what's missing is the concomitant motion of an RNA helix that helps us to take over that position where EFTU should be. We also find isolated 50S particles. We're interested in figuring out whether these are assembly intermediates and we can learn about them. Then we saw this weird grouping of particles where there's new density that's sort of at the end of the box. And Barrett looked at this and he's like, I think something's there, but it's getting like cropped out by the box. I don't know what's going on there. What's neat about this is we can just go back and say, well, let's just look at a bigger box. We have the whole cellular context. What else can we see? Let there be light. What else can we see? Okay, so he trains a new TomogrDragon model on this larger box. And now we start to see some really interesting structures. So we see things like a ribosome bound on, this ends up being a lipid bilayer. we go and look at these particles, we can map them back to the cellular context, and they all map to the plasma membrane. So, we weren't looking for these things, it just naturally falls out of the data, but you can find co-translational translocation directly, and I won't show you all the model building for this, but we can actually see elements of the sec translocon on the distal side of the membrane, and actually see polypeptide being translated there. What are some of these other classes? You'll look at these and be like, oh, what are these blobs? We also asked, what are these blobs? So we went and looked at each one of these structures. Turns out the distance between the ribosome that we could resolve and the blob was tightly distributed for 3 of the classes, and for these other things that don't have the blobs, there was a broad distribution. And this distance was interesting because it's about the distance between 2 ribosomes that are on a polysome. So when most cells translate things, ribosomes sort of stack up next to each other and they're co-translating the same mRNA. That's about the right spacing. And then if you look at the relative orientation of the 2 ribosomes to each other, you can see that they're highly stereotyped for these particles, but not for others. Which probably have to do with them both binding to the same. We can go back to each one of these particles and now generate higher resolution structures for the other blob, again using CryoDragon. We can place those into the cellular context, so each one of these is now a different difilm. If we go and look closely at those, we can actually find the bridging mRNA. So I don't know how much people know about how translation works, but the way it basically works is that there's an mRNA that's going to come through here. These tRNAs are going to decode the mRNA, perform a peptide transferase reaction. That's where the peptide comes off. So this is the peptide transferase center for one ribosome. We can find bridging density that now goes to the peptide transferase center for the next ribosome. So we can actually see mRNAs that are bridging. There's other ones that are close, but clearly not connected. So there may be mRNA there, there may not be. I'll just sort of leave you with sort of what we see. So the movie here of all the heterogeneity amongst these disomes. Some are very close together and tight, some are further apart, and then some are completely distal. Bridging mRNA just happen to be. Okay, so with that, I think I'll stop. I think I'm about on time. Go ahead and stop and thank the folks who did this work. So again, Ellen John sort of started all of the CryoDragon work, heroic effort in Princeton. Eric Howell did all of the graphic work. Really fantastic students, graduated this year. Will Kimmin did all the work. Graduating. Rest of the time taking questions.
You showed a nice images of the latent space training an autoencoder. And then you showed a video of drag across the latent space. You can see this movie. It seems like you could drag almost
any direction.
And I was just wondering what you think about that. Like, how do you, if you're going to show a movie, you must pick a path.
Yep.
You're not going to pick every possible path. So how do you?
Yeah, I think there's sort of 2 interesting questions there. One is that there's no good reason to think that the latent space is going to be organized along any interpretable reaction coordinate. So just looking along latent Z0, I don't know why that should be interpretable. That being said, within the latent space, we have just a graph traversal algorithm where we say if you see 2 states that you're interested in, pick those as the endpoints, and then we'll just use Dijkstra's algorithm to find the shortest path between them using data throughout it. So that's a reasonable approach. What that means about the actual intermediates, I don't want to say. I think the more interesting thing is to think about how do you go from this uninterpretable low-dimensional space that you get out of the autoencoder to something that is actually interpretable molecularly. And the way we're thinking about that, and I'd love to hear other people's thoughts, but the way we're thinking about that is to basically generate a ton of volumes. So maybe you sample, you oversample, or you sample extremely extensively. So you have 100,000 volumes. And then you just do PCA on the volume space, because now those are in real space, sort of molecular, structural changes that are organized by their variants. And so now you can start to look along the reaction coordinate that's the first principal component. And when we've done that, we've seen that that's like the dominant mode of motion. And then the next principal component gives you some second motion. The downside of that is that it's this linear combination. And in reality, these things may not be linear combinations. And so there's some motions where we know the ground truth is like complicated motion. I mean, you could sort of see it in that rotary motion I showed you, where you saw that the traversal was like this big C. That's almost certainly because we did PCA on the latent space to be able to get that. And so again, those you can involve variables to explain a complex nonlinear motion.
Incorporating any kind of assumptions or priors, because it looked like those motions seemed very natural. So that
the only
so
it's a variational autoencoder. So the only regularization is that the loss that you calculate is it's two terms. One is the sort of reconstruction loss, which is the difference between the two images, and then there's a KL divergence loss, which is the difference between the unit Gaussian and then where you project. So you're sort of penalized by pushing things apart really, really far in latent space, and so that gives you some degree of regularization that makes these things look much smoother. It sort of helps with some of the overfitting. Yeah, we're like regularly surprised by how often the latent space is organized by structure. Again, there's no theoretical. The one thing is that like we typically train on, I guess for autoencoders it's relatively low dimensions, but for us to think about it's high. So it's usually like an 8-dimensional latent CryoDragon or 128-dimensional latent for tomography. Either way, to be able to make these plots, we have to do some dimensionality reduction. And so we take those and we often use work well with this data, but then of course all the distances are total. Yeah.
I was kind of curious for, I guess, CryoDragon and also the tomography. Each time you run it, do you get like a different space? How consistent is it from like run to run?
Yeah, I should. I don't know if you're a reviewer too, but reviewer 2 asked a very similar question. It is in the supplement of the NSMB paper. I got that question. I was like, oh God, this is going to derail this whole paper. And then we did 3 replicates with 3 random seeds. They were like, okay, there were some inversions of an axis, but otherwise they were like superimposable. Very surprising how that was. Well, it was a very thoughtful review. Yeah. Yeah. Yeah, like some sort of dynamic boxing or something. I think this would be cool. So there's sort of 2 parts that you use to decide the box. One is that typically the micrographs you have to pick particles from. And when you do that particle picking, you often have to sort of define a box size that you think is appropriate. And then based on the reconstruction, it'll be going to the edge of the box. And so then you have to make the box bigger or something like that. We played with, I had a Europe student who played with this and love it if someone else took this idea and got it to work, because we couldn't get it to work. But the idea was to try to use some of the segmentation algorithms that don't rely on boxes. So there was this, it was like, this work. There's like a diffusion box picker where it sort of has dynamic scaling of the boxes, and we tried to use that as a particle picker where you could get all types of different particles, and it just like never really went anywhere. But people are doing this for image segmentation where you have like a big horse and a little rider and you want to pick those separately, so you have 2 different size boxes. So we're trying to adopt some of that. Yeah.
So we know that this is experimental how do you take these
Yeah. Yeah, I think it's the key question. So I still see structural biology as a hypothesis generating tool. Feeding structures. They allow you to generate hypotheses about specific contacts, for example, function. And I think that the onus is on the experimenter to then say, okay, like other tools, for that interaction, for example. On using hypotheses, doing those experiments, seeing what the new function is, and seeing what the change structures are really required.
question is about, so you're extracting these ensembles from this data, and my question is what do you consider as a ground truth or a benchmark, whether it's you comparing to another set of data or how they determine that it should be a ground truth?
Yeah, so there's two approaches, there's three approaches. So the first approach is that you simulate data and you have ground truth labels. That's great because you have ground truth labels. The problem is that the simulations are never right. They don't capture the noise model properly. There's all these imaging artifacts that come through in real data. And we have built things that work great on simulated data and not on all real data. We've also built things that are on simulated data that work great on real data. So there's a lot of reasons that I don't love the simulated data. The second thing you can do is more or less what I've shown you today, which is you can work on systems for which there is a lot known already. If you're trying to develop these algorithms, and then you can basically rediscover biology, which has been validated by other means. The challenge of that is that you send these papers out for review, and one of two things happens. You either recapitulate what people know, and the reviewer says, we already knew that, we don't care, or you find new things, and the reviewer says, I don't believe you. And so I'm sort of with this conundrum. So the third approach is something that we've been working on, which is to sort of make biochemically defined ground truth data sets. And the way that we've done that is we've taken a dead Cas9, so catalytically inactive Cas9, that gives you sort of ability to target a DNA of interest, and synthesize that DNA, and then we can just make it with various numbers of nucleotide extensions from the Cas9. So you have single base pair resolution. And so you can biochemically purify, say, a dozen different complexes, each with an additional base pair, image all dozen of those separately. You now have ground truth, you know, all of them are the +1, +2, +3, blah, 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 blah. And then you can take those particle stacks and then mix them in whatever distributions you want, but you still have the labels associated with the images. And then try our algorithms or anyone else's, and basically just ask, like, who gets the distributions right? And when they're wrong, which types of particles are they failing on? And are there certain pathologies associated with those particles that cause them to fail? Is it always from this viewing angle, you can't figure it out? Or is it that this particle is exposed to too much dose?
If you take your
not very well. Yeah, so it turns out it's terrifying results. So actually CryoDragon does reasonably well with it. The 3D classification tool that people often use, and this is maybe one of the things you asked me if I wanted to not record everything. I'm going to say something now that maybe we can cut. But we've tried a number of the competing tools. We're not expert users of them, but you draw the confusion matrix of your ground truth versus the predicted labels, and it's just like a flat box. And so that's very concerning for all the traditional classification. And I think what that's telling you is that the genetically encoded heterogeneity of these one base pair extensions is relatively small to all the rest of the imaging. Right, there's other things, there's other signals there. It's not that the algorithms are like wrong. They're right in that they are classifying some feature of the image, but it's not the feature that we care about. And so you can do things like you can put a mask if you know where the changes are, and then the algorithms do much better. So we think that the underlying data set is correct. But yeah, the out-of-the-box tools are worrisome when you look at the results. Yeah. Sorry, I couldn't hear the beginning of the question.
Oh, can you use the presence of like functional domains based on analysis to somehow bias or solution?
Yeah, there's it's an interesting idea. So there's sort of two schools of thought on this. One is that two schools of thought on this in terms of structure determination. One is that you have your underlying data. That data allows you to generate a density map. And into that density map, you build an atomic model that's most consistent with the density map. And there's sort of this firewall, at least in cryo-EM, between the density map and the atomic model. And I think that there is some value to that. The downside is that if you know the protein you're looking at, you know its domain architecture, why not use that inductive bias to help you generate a better map? And so we and others have done a couple things where you try to, for example, directly infer the atomic model from the particle images. And there you can imagine using priors based on sequence to structure predictions to help guide that. And I think the danger is that you overfit to your model and you sort of have this self-fulfilling prophecy of getting back the structure that you gave it, independent of the data. And so it's getting that trade-off right, but I think that there's promise there. Something we haven't spent a ton of time on, but worth thinking about. Yeah. Yep. Yeah, so I think, you know, the stuff that we worked on are all large domain motions. Like even that helix moving, right, that's probably like 8 or 10 kilodaltons of mass moving. And it's quite a large signal. I think that this example of this this encoded heterogeneity on the Cas9 is a little bit closer, and that's really the limit. So there, you're adding on the order of like 600 daltons of mass with each base pair that you add. And I would say we're pretty good at like 3 base pair deltas. So there you're on the order of like 1,800 daltons. We're less good with 1 base pair deltas. So it's somewhere in that range of like a kilodalton. Mass as the detectors get better and as these algorithms get better, that's when you get pushed lower and lower.
One question from all your maps seem to be of low resolution. Images in the manifold, images in the computational bin, which are actually different.
Yeah, it's a great question. So I would say that is true in general. The maps that we generate are low resolution relative to what you can get if you just focus on the structurally homogeneous particles and basically isolate those to increase your averaging capacity to push the resolution. And that's a conscious choice. Like you sort of tune that dial however far you want to go based on how much data you want to throw away and how homogeneous you want things to get. The other thing I would say is that what we often do is we sort of use CryoDragon as a classifier. So we sort of get a bunch of low resolution maps, but then we say like, okay, this set of particles we're actually interested in because they're all largely in the same conformation. And we have some tools now to basically extract all the particles that are in that conformation. And then we can take them back through traditional reconstruction methods, which have a bunch of bells and whistles that we haven't built in. And now you can start to push the resolution much more. But you should never expect to see these sort of like sub 2 angstrom maps that people get of ferritin if you have a complex that's highly dynamic. It's just not going to average. Yeah.
What's the state of the art for imaging solving structures by cryoEM or CryoDragon for small molecules? You showed, you know,
yeah.
medical. And I'm just wondering if it's sort of the density there sort of looks also like a kind of blob. I
yeah.
could have modeled that in probably any which way.
For sure.
And I'm just wondering, is there could one use something like this and really, if you were really interested in small molecule binding, sort of get out all the binding modes that depending on the resolution?
Yeah, I think this gets back to this question of like how small of a signal change do you detect? And so if you had subtle changes in conformers, like let's say you just had 2 different conformers that could bind, could you sort on that signal? So I've restated your question, but haven't given you an answer. We haven't been able to do that. I think the closest thing that comes to mind is that there's this really nice workflow from Thermo Fisher and Basil Gerber's lab looking at a series of like 24 small molecule inhibitors of some pharmacologically important target that's on the order of 100 kilodaltons, whose name I should know, but it is escaping me. Anyway, but the way that they did that is that they determined separate data sets with each small molecule bound, and then each one of those data sets goes to sub 3 angstroms, maybe sub 2.5, and then they can nicely see one of the compounds bound and what conformation it's bound in. That's not sorting heterogeneity computationally within a data set. It's using physical additions of the material, sorting those on separate grids and imaging them separately. But they're seeing these beautiful maps of a ton of different compounds. So I think people are starting to think about cryo-EM as a method to screen for where things bind. I'm not in pharma, but like more and more people in the groups of pharma are starting to think about this. And I think it's, or maybe now where it's like, depends a little bit on like who's running the group and what their biases are. And I think that's like a testament that cryo-EM has caught up. I would say like if you have proteins that crystallize and you can soak, that still seems to me like it's going to be faster to just like go through a compound screen. If we're getting close. Yeah.
Other two questions. It looks like you picked like the protein that is the most abundant in the cell. What would be the challenges like getting all the rest of the protein to capture those guys?
Yeah, I think it's there's two problems. One is that you need to be able to pick them initially, and there's not a lot of signal to pick on. So you need things that are big that give a lot of signal. Ribosomes are great. Proteosomes are great. There's probably a few other targets there. And then the second is that the throughput of tomography currently is quite low, which means that you don't get that many tilt series, which means that if, even for CryoDragon or TomoDragon, you still are averaging across particles in the same sense. And so that means that you need relatively abundant complexes to get up to the numbers that you need if you have 100 tilt series or something. So that's starting to change. Thermo has a new instrument that should be able to generate these lamella in a relatively high throughput fashion. And so if there are donors in the room with $6 million, I'm available. I'd be happy to talk about buying one of these instruments that I think are going to push throughput. No, but I'll just say, I think the way this is going to go is that those types of instruments will end up in like national centers. And then you'll be able to send some cellular samples and some condition you care about. They'll be able to collect a few hundred tilt series, like 1,000 tilt series for you. And then that's just going to basically proportionally push the particle count down in terms of the lower band expansion. But right now, it's great for ribosomes. We happen to work on ribosomes. We think it's a well-matched system. Other things. Oh, yeah.
So you image, are you looking into bacteria then?
So you can do some, you can do some human cells. You still just need to cut this lamella into them. Okay, there's a trade-off there. Advantages that there's a bunch of machine biology. The downside is that you still have a you think about you have a fixed volume that you can image. And if you want to sort of think about, I want to see a representative sample of the cell. In bacteria, you could maybe convince yourself that that's the case because they're so small. You get to a huge neuron or something like that. You're looking at like one street corner and saying that that's representative of all Boston. Yeah, in the back. Yep. Yeah, this is a good question. I mean, the challenging thing is that definitionally the transition states are unpopulated. And so we still only see what we see. So we're still seeing the energy wells and not the maxima. And so, so I think that the where this could be useful is if you can see within a data set substrates and products, then maybe you can use that as endpoints for MD, and then let the MD try to get you across plausible transition states. But they're not, I mean, they're lowly populated, we're just not going to see them. Yeah. Yeah, it's a cool idea. We're not doing any of this, but the variant of it that sort of comes to mind is that you can add gold nanoparticles that give you an insane amount of contrast. You can conjugate those to antibodies and then use the antibody as an affinity reagent to label your particle of interest. And the challenges there is like delivery, and do you have to lyse the cells first, or how do you get them in? But But I think that it's like a super cool approach because, like you say, it basically just puts like a big flag post and says, hey, I'm here. And then you can use all the antibodies that we have for defining this.
Well,
Thank you guys.
can Joey one more?
Finishing the data again next month.
That's right, yeah. Next month, we originally scheduled for the 8th, we're going to do the 15th.
Well, I realized I didn't repeat any of the questions. Sorry.
I've been typing them.
No, good. Somebody's competent. Appreciate it.
I tried. I made a lot of
Awesome.